Hello and welcome to the next lecture in our Anatomy and Physiology course. This lecture is on the lymphatic system, commonly known as the immune system, and the material can be found in Chapter 21. What I'm going to be talking about is I will start by covering the components of the immune system. What are the cells that comprise the immune system? I will talk about a process which is critical to the immune system called phagocytosis. The word phagocytosis simply means cells eating things, usually immune system cells eating things. So if a foreign bacteria gets into your system, probably one of the things that will happen to it before it can, if you're lucky, before it can infect you, is it will be eaten and therefore destroyed by a phagocyte. So phagocytosis is the process of immune system, white blood cells basically in your body eating bad guys that get into your circulatory system. Inflammation, you know what that is? That's when you have a body part that gets abused somehow. Either you hit it or it gets infected or you get a bee sting or something like that. And inflammation is characterized by swelling, pain, redness, and a sensation of heat. And we'll see that that is actually kind of a slightly inappropriate immune system reaction. But it is an important one, so we'll talk about that. And finally, part of something called the humoral immune system is the production of antibodies. So you know that, for instance, cold viruses ultimately are destroyed by our immune system rounding up something called a B cell, telling the B cell to turn into something called a plasma cell. Turning into is the technical term, is to differentiate into. So a B cell will differentiate into a plasma cell. A plasma cell is specialized to kind of shoot antibodies at a foreign invader and destroy it. So antibody production, the response to a foreign invader entering, like a bacteria or a virus entering your body and being destroyed, is usually or often accomplished by plasma cells firing antibodies at it. We'll talk about the different white blood cells that constitute the immune system and what their different functions are. Of course, the word leuko means white, so leukocytes is a general term for white blood cells. But as we learned in the last lecture on blood cells, the fact is that there are other types of white blood cells besides lymphocytes. There are two types of white blood cells. There are leukocytes, which leukocytes is the general term for all of them. Lymphocytes belong to the lymphatic lineage, and granulocytes are the other type of leukocytes, and they belong to the myeloid lineage. All right, we'll talk about the difference between the innate immune system and the humoral or the adaptive immune system. The word innate means that you're born knowing how to do it, and we have certain white blood cells that are born knowing how to recognize and instantly destroy certain types of foreign invaders, to destroy certain types of bacteria usually. And that is and usually they respond to certain types of bacteria by eating them. So these are usually phagocytes. So phagocytosis and the phagocytes that do this are part of the innate immune system, or what's known as the cell-mediated immune system. In contrast to that, we have something called the humoral immune system, also known as the adaptive immune system. The humoral immune system is referring to the production of antibodies by plasma cells, and that is a much more delicate, much more specific way to kill foreign invaders like bacteria and viruses. And it is called the adaptive immune response because your immune system is not born knowing how to destroy these viruses. It has to adopt. It has to learn what they look like first of all, and then after having learned what they look like, it has to go and find a B cell that can produce antibodies that will destroy it. So it has to adopt. So we call that the adaptive immune response. Okay, now we mentioned earlier about inflammation. The word chronic, of course, means constant or long-term, and chronic inflammation is not a good thing, because if a particular part of your body remains inflamed for a long period of time, you develop something called fibrosis, which is a fancy term meaning scarring. To put it in a very simple way, it refers to scarring. So if your body is if a particular part of your body is inflamed for a long period of time, that body part will, the immune system is kind of not only destroying foreign invaders or something else, but it also has the effect of damaging your body. And when your body is damaged, one of the ways it tries to heal itself is by forming scar tissue. We learned in the section on the integument that scar tissue is not really the same. It's a type of tissue that's not really the same as it is replacing. It tends to, scar tissue, when scar tissue replaces epithelial tissue, for instance, scar tissue contains more connective tissue and fewer cells than does the tissue that it replaced, and it leaves a scar, which is obviously quite different than the tissue it replaced. All right, finally, the lymphatic system is not just the cells. It also refers to a network of vessels that are sort of like veins that run around all over the body that are referred to as lymphatic vessels. Basically, they act, among other things, they act as a kind of a backup drainage system for an emergency drainage system to remove surplus excess fluid from interstitial spaces. The word interstitial means between cells, and so there are, in the human body, we have types of tissue that are called soft tissues. Adipose tissue is a good example of a soft tissue. And in soft tissues, the cells are not tightly packed together the way they are in epithelial cells. They are loosely packed together, and there's space in between. And that space in between the cells is usually filled with fluid. The space in between cells is called interstitial spaces, and then the fluid that occupies the spaces between cells is called interstitial fluid. And if you allow too much fluid to build up and accumulate in between these cells, you get swelling. And the technical term for swelling is edema. Right? So the lymphatic system, one of its main functions is to drain away excess fluid from the interstitial spaces to prevent swelling. Where did that excess fluid come from in the first place? It usually came from serum leaking out of capillaries. So some capillaries are deliberately leaky, and some of them are just accidentally leaky, or they leak some of the time. But in either case, you do have serum leaking out of the capillaries into the interstitial spaces, and then it is drained away by the lymphatic vessels. Now, once that serum gets into when the serum is in the blood, we refer to it as serum. When the serum leaks into the interstitial spaces, we call it interstitial fluid. And when the serum is taken away by lymphatic vessels, we call it lymph. So the lymphatic vessels carry a fluid that is called lymph, but it originally was derived from the serum. Now, this is kind of cool because that fluid is then drained back into the bloodstream at the level of the subclavian veins. Right? So the lymphatic system drains it drains the serum back into the bloodstream because it is connected to the subclavian veins. But before it does that, the lymphatic system gathers up all of this lymphatic fluid, and before it gives it back to the bloodstream, it checks it for the presence of invaders like bacteria and viruses and even cancer cells, because all of these lymphatic vessels are punctuated by little tiny organs known as lymph nodes. The lymph nodes are little tiny filters that are filled with white blood cells that check the returning lymphatic fluid for bacteria, viruses, and cancer cells. And if they find any in there, they multiply and gang up on the invaders and destroy them. And because these white blood cells that are inside the lymph nodes multiply, what happens is that the lymph nodes will swell up. They will swell. And so one of the indications that you're sick, one of the ways that a doctor tells whether you're sick or not, is to feel your lymph nodes. And there's a lovely set of lymph nodes underneath your chin, underneath your mandible. So those are called the submandibular lymph nodes. And there's another set in your armpit. And of course, the armpit is known as the axillary region. So there's a set of axillary lymph nodes and a set of submandibular lymph nodes. And when you go to the doctor's office and you say, I feel sick, the doctor will feel underneath your chin and then feel underneath your arms and see if the lymph nodes are swollen. And if they are, that's an indication that your body is trying to fight something off, some kind of an infection or perhaps even cancer. Women know very well that when they do a breast self-exam, one of the things you do is you rub yourself under the armpit to check and see if your lymph nodes are swollen, and if they are, that could be an indication that your body is attempting to fight off breast cancer. All right, and then of course, as always, we'll finish the lecture off by talking about some of the pathologies of the immune system, some of the things that go wrong. All right, let's begin. What is the immune system's job? Okay, so the immune system's job is to destroy foreign invaders that enter the body, including bacteria, fungi, protozoa. Protozoa are small, they're actually quite large unicellular eukaryotes. Parasitic worms like nematodes also invade your body, and if you're lucky, the immune system destroys them, provided that you have a functional immune system. And one of the amazing skills that the immune system has is it has the ability to distinguish your tissues from foreign tissues. It has the immune system is quite good at distinguishing your proteins from foreign proteins. It's a little bit less good at trying to distinguish foreign carbohydrates from your carbohydrates, and has a bit of a difficult time trying to distinguish foreign lipids from your lipids, but it's very good at distinguishing foreign proteins. So the proteins that you would find on the surface of a virus are different than your proteins, and so your immune system is able to look at viruses and say, you're not supposed to be here. You're an invader, and then they will destroy it. Your immune system will look at proteins that are on the surface of a bacteria and say, "You're not. I recognize you. You don't belong here. You don't look like any of the proteins around here. You're a foreign invader," and they will destroy it. Right. So our immune system is very good at recognizing foreign proteins and destroying them. Right. So now a foreign protein that provokes the formation of antibodies. We just mentioned the fact that there are certain white blood cells called B cells that can differentiate into a type of cell called a plasma cell. A plasma cell is a specialist cell whose job it is to secrete antibodies that bind to and destroy foreign invaders. Right. So any foreign protein on an invading cell that is capable of causing the production of antibodies by your immune system is referred to as an antigen. Right, so anti refers to antibodies, and then gen refers to antigen, the cause of, or the reason why they were generated. Right, so you have foreign proteins. We sometimes use the term antigens or foreign antigens synonymously with foreign proteins. All right, now the innate immune system, which I mentioned earlier, right, the innate immune system, there are some leukocytes that are automatically able to recognize certain foreign antigens, and they are part of the innate immune system. There are other leukocytes that have to be told how to attack a foreign antigen, and they are usually told to do so by a type of a white blood cell called a helper T cell or a T helper cell. Here's the abbreviation for a T helper cell. Right? And they are part of what's known as the adaptive immune system. Right? So you can imagine that the adaptive immune system is kind of like an army, and they will not attack somebody unless the general tells them who to attack first. Right? So the general then has to go and see who's out there and what they look like, and then come back and tell the army to attack a certain gang of invaders that are wearing a certain type of uniform that the immune system is able to recognize. Okay, so because the T helper cells have to learn to distinguish foreign antigens from your antigens, one of the common sayings is that T helper cells learn how to tell the difference between foreign antigens and your antigens. They learn how to do this by going to college, and the college that they go to is they pass through an organ called the thymus. The thymus is located right above your heart, and when we talk about we're talking about the lymphatic system today, but when we talk about the endocrine system producing hormones, you'll hear about another organ called the thyroid. Do not confuse the thyroid with the thymus. A lot of people do, and they get marked deducted on final exams. The thyroid is part of the endocrine system, and it produces hormones. The thymus subject of today's lecture, is part of the immune system. And what the thymus does is it's a place where T helper cells go to learn how to distinguish the difference between foreign invading proteins and your proteins. Okay, now in fact, they don't actually learn anything. That's kind of a metaphor. That's an analogy. The truth is that what happens is all of the T helper cells are sent through the thymus, where they get a sort of a test as to whether or not they can distinguish your tissues from foreign tissues. And if they fail the test, they're destroyed. Right, so this is kind of the equivalent of Columbia College. We don't teach you any biology. We just let you in on we bring you into a class on the first day and give you a biology test, and anybody who doesn't know the material already is destroyed. Right, so that's not really education. That's selection, which is what's happening there. Okay, so the thymus is important in making sure that the T helper cells are able to recognize foreign antigens from your proteins, and it does this through a selection process. All right, what is it about your proteins that the T helper cells are recognizing? Okay, there's a complex of genes, a whole bunch of genes, a whole collection of genes that code for a bunch of little proteins that are found on the surface of every one of your cells. And those little proteins are, depending on whether you're a biologist or a medical doctor, those proteins are either referred to as the major histocompatibility complex, also abbreviated MHC complex, or they are also known as the human leukocyte antigen complex, or the HLA complex. It means the same thing. And these two, this cluster of genes, it's a group of 600 genes that are clustered onto 3 different groups on chromosome 6 of the human genome. And because they're clustered into 3 groups, we refer to them as MHC class 1, MHC class 2, and MHC class 3. Right? But we're talking about 600 genes that encode 600 little tiny proteins that are present on the surfaces of your cells. And the presence of these proteins allows T helper cells to recognize the fact that any cell that has those proteins on the surface is you and not a foreign invader, and should be left alone. If certain cells in the immune system encounter a cell that doesn't have those MHC class proteins on the surface, it will say, okay, this is not supposed to be here. This is not part of me. I'll destroy it. And so that's what they do. Sometimes the immune system makes a mistake and it misreads it. There was a problem educating the T helper cells in the thymus or something like that, and your immune system ends up destroying your own cells by accident, and that is referred to as an autoimmune disease. Autoimmune diseases are very common, and we'll talk about those when we get to the pathology section. Okay, when you're doing tissue transplanting, you're transplanting a heart of one person's heart into another person's heart. We already know from the previous lecture that you have to match the blood type. So you can't go around transplanting a type B heart, the heart from a person with type B blood into the body of a person with type A blood, because the heart would be rejected by the type A person's immune system. But once you've matched the blood type, your job is not finished. You also have to match as many of the MHC class proteins as you possibly can, and that's not easy. Generally speaking, the closer a person is related to you, the more of the MHC class proteins match. And the only person who has exactly the same MHC class as you do is your identical twin sibling. So identical twins can donate organs to each other without any problem at all, whereas uterine twins or maternal twins or fraternal twins can only, they can donate tissue, they can donate organs to each other, but they may be rejected a little bit by the body's immune system. And so somebody who has an organ implanted into themselves that came from somebody else, even if they're a close match, they have to have their immune system suppressed by drugs, because otherwise their body would reject the transplanted organ. All right, let's look at the components of the immune system. To begin with, all of these B cells and T cells and granulocytes and leukocytes just like all of the other blood cells, came initially from the bone marrow. And we learned about the fact that there are two lineages, the myeloid and the lymphoid lineage. Okay, the word leukocyte, of course, means white blood cell, but there are two types. There are the granulocytes, which are white blood cells that come from the myeloid lineage. They're called granulocytes, of course, because they have little grains. They have little vesicles inside their cytoplasm that are filled with whatever it is they use to do their job. And so under the microscope, they have a grainy appearance. And the lymphocytes, for this course, which is an introductory course, the only lymphocytes we're really going to care about are the B cells and the T cells, but there are others. Okay, the thymus is an organ that sits on top of the aorta of the heart. It's a kind of a squishy, mushy organ that looks like, if you see it, it looks like a blob of fat. In Biology 120, we have students dissecting rats, and when we were doing the cardiovascular system, the students open up the thorax of the rat, and then the first thing they see is the heart, and above the heart is this jelly stuff that looks like it's fat, and we always tell them, look, take a good look at that, because it's not fat, it's the thymus. And then, if you're going to look at the aorta, you have to remove it, but just remember, take a look at the thymus first, because get a good look at it, because that too is a very important organ. It's not just a blob of fat. And the purpose of the thymus is to educate or screen the T helper cells to make sure that they are not going to start destroying your tissues instead of just foreign tissues. The lymphatic vessels are a network of vessels that are about the same size as veins. In fact, they look a little bit like veins, and they provide a backup drainage system for the interstitial fluid. And those lymphatic vessels are punctuated at regular intervals by little tiny organs called lymph nodes that check the lymphatic fluid for bacteria and viruses on the way through. The spleen is in fact a giant lymph node that checks the blood for invaders. And from the last lecture, we also learned that one of the main purposes of the spleen is to destroy and recycle the components of worn out blood cells. There's some other immune tissues, the tonsils and so on, the appendix. We're not going to really talk about those in this course because this is only an introductory anatomy course. Okay, so ultimately all of these cells come from the bone marrow. If you look around for figures about the immune system on the internet, you'll often find this figure, but this figure, I wanted to point out to you, this figure is completely wrong, because this figure, while there are a lot of good things about it, it's showing that the blood cells come from, they're showing blood cells coming from this bone. Okay, number one, what bone is that? You can pause the video and then see if you got the answer right. And then, okay, so the answer, of course, is that is the femur. Okay, and then the next answer, my next question to you is, which bones do the blood cells come from? In an adult, in a baby, you do get blood cells from the femur, but in an adult, really the only place that you would get blood cells from a femur is the head of the femur, which is made of spongy bone, trabecular bone, and you would certainly not get any blood cells coming out of the shaft, because that's just filled with adipose tissue. Okay, so this figure is a little bit wrong. This figure is better because it doesn't show you that, but this figure does show you the location of the thymus, shows you the location of the spleen, shows you the adenoids, which are in your nose, which is part of the immune system that's associated with your nose, shows your tonsils, the tonsils are actually they are part of the immune system. It's showing all of these lymphatic vessels, this network of vein-like vessels that are carrying lymph back from the bottom of the body upward and then dumping it into the subclavian veins. And so it's showing the submandibular lymph nodes, and it's showing the axillary lymph nodes here. But the network of lymph nodes is very extensive throughout the body. All right, so the bone marrow is the source of all the blood cells. It is where hematopoiesis takes place, and the cells that carry out hematopoiesis are called hematopoietic stem cells. They are pluripotent. Last time we learned the difference between pluripotent and totipotent, and they differentiate into a number of different white blood cells in response to specific cytokines, like erythropoietin. Do you remember what that does? You're supposed to. Interleukin 2, interleukin 3, MCSF, and so on. And the source of those bone marrow cells is the flat bones, the red bone marrow. So this is where they actually come from. And so we saw this figure last time. These hematopoietic stem cells will, in response to certain cytokines, they will differentiate into the myeloid lineage where they can become any of these, or the lymphoid lineage where they can become any of those. All right, there are 4 types of white blood cells that can that attack invaders. Okay, how can they? Sorry, 4 types of attacks that white blood cells can make on invaders. All right, so number 1, some of the white blood cells are able to physically eat the invaders, right? And we call those phagocytes. Phagocytosis is the process of cells eating things. There are several different types of white blood cells that are classified as phagocytes. They are called monocytes, neutrophils, or macrophages. They are all phagocytes, and they're given different names mainly because you find them in different parts of the bodies. But on the final exam, I could ask you to give me 3 examples of phagocytes. Give me 3 examples of phagocytic cells. There are some cells whose job it is to produce antibodies. Antibodies are a very subtle, specific attack on a specific foreign virus or foreign bacteria. The antibodies are produced by B cells, but not until the B cells have differentiated into a related cell called a plasma cell. Right, so you know what a lawyer is, right? So a lawyer ultimately does the law, but not, a law student is not a lawyer, right? So B cells are kind of like law students, and then when they pass their exams for the bar, they become a plasma cell, and they're able to fire antibodies at criminals to destroy them. Okay, there are some white blood cells that attack foreign invaders with proteases. Remember, a protease is an enzyme that hydrolyzes protein. So pepsin in your stomach is a protease. There are other proteases in the human body that we'll talk about later. But there are white blood cells specifically called eosinophils, and their job is specifically to attack microscopic worms by dumping proteases on them. The worms, of course, are made largely out of protein, and so that they'll be destroyed that way. And there's some white blood cells that destroy foreign invaders or help to destroy foreign invaders by causing inflammation. And what they do is, okay, so imagine that you have a bad guy, you have a bacteria or a virus in the soft tissues, in the adipose tissue. Where are your white blood cells who are supposed to destroy it? Well, they're stuck in the blood vessels. They're stuck in the capillaries where they're not much use. So the white blood cells are of no use to you if they are inside the capillaries, and the bad guys are outside the capillaries in the fat cells, in between fat cells. And so what inflammation does is there's some cells that release chemicals that cause the capillaries to become leaky. And they become leaky enough that the phagocytes can get out and go after the bad guys. And you know that white blood cells are a little bit bigger than red blood cells, so you know that if the phagocytes can get out, then the erythrocytes can get out as well. And that's why, and so can a lot of the fluid. And so that's why when you do this, when you make the capillaries permeable, the word permeable is a fancy word for leaky. When you make the capillaries permeable, the area where the capillaries became permeable swells up because a lot of fluid is leaked out. They turn red because they now contain erythrocytes. Erythrocytes have leaked out of the capillaries into the soft tissue. And they feel hot because the white blood cells are releasing a bunch of hormones and cytokines that have the effect of making you feel hot in that area. So blood cells, white blood cells that are involved in causing inflammation include cells called basophils, mast cells, and the mast cells make the capillaries leaky by releasing a chemical called histamine. So mast cells are granulocytes, and so if you look at them, they're grainy. And what the grains that you see are, the grains are actually the grains are actually little vesicles that are filled with histamine. Okay, so you know that sometimes your body does this and releases histamine when it's not supposed to. It happens in hay fever. And so what kind of pills do you take if you have hay fever? You take antihistamine tablets. And so those antihistamine tablets are making it more difficult for the mast cells to release their histamine. All right. There are two types of immune responses to foreign invaders that immunologists like to think about. One is called the cell-mediated immune response. One is called the humoral-mediated immune response. Cell-mediated immune response is where cells like phagocytes directly attack the foreign invader. Eosinophils will dump proteins onto worms. Macrophages will eat bacteria to destroy them. And because that particular immune response is controlled or mediated by cells, we call that the cell-mediated immune response, or a cell-mediated attack. The word humor actually means liquid. Right? So sometimes in anatomy and physiology you hear the word humor or humoral referring to liquid. It doesn't mean laughter. It means liquid. That's the original meaning for that word. So the humoral immune response is where you have B cells differentiating into plasma cells and then sending out soluble antibodies. So when a virus or a bacteria is infecting the body and plasma cells are shooting antibodies at it, those antibodies are made of protein, and they are globular proteins, and globular proteins are soluble in water. They are soluble in aqueous solutions. So because we're attacking the foreign invaders with soluble proteins called antibodies, and we're talking about liquids, that's why this is referred to as the humoral immune response or humoral immunity. As I said earlier, cell-mediated immunity is usually immediate because it is part of the innate immune response, meaning that the cells that do this, the phagocytes whose job it is to eat bad guys, they are born knowing how to do it. They don't have to learn how to do it. The humoral immune response takes about 2 weeks because the cells that are involved have to learn how to do it. They have to learn what the enemy looks like, and then they have to do a bit of training, and then they go out and attack them. And so the humoral immune response, which involves soluble antibodies in liquid, soluble in liquid, takes a couple of weeks, and therefore it is referred to as the humoral immune response, but it is also referred to as the adaptive immune response because it has to adapt to a specific foreign invader. Okay, so as we said, the innate immune system is where you have these phagocytes and other cells, phagocytes and eosinophils and other things that know how to destroy, and they are born knowing how to recognize certain bad guys and attack them immediately. Now, when you have a cut and it gets infected, it often develops this kind of smelly, sticky stuff underneath the bandage that's called pus. Right? What pus actually is it's the remains of millions of dead phagocytes that have swarmed into the area to attack and destroy an invader that got into the cut. Right? Now there are so I said that these cells are born knowing how to recognize and attack certain invaders. An example of the invaders they are born recognizing and knowing how to attack is a bacteria called Staphylococcus aureus. And so whenever Staphylococcus aureus is normally present on your skin, and if you get a cut, the Staphylococcus aureus may go from your skin into the tissues underneath your skin and immediately provoke an immune response from it will provoke an innate immune response immediately, causing these phagocytes to swarm to the area and try to eat the Staphylococcus aureus and forming pus in the process. Now that process of forming pus is referred to as a pyogenic response. Okay, so there are some bacteria that, this is not so critical for this course, but if you take the microbiology course, there are a whole bunch of bacteria like Staphylococcus aureus that are classified as pyogenic bacteria because when they get inside your body, they provoke the formation of pus, and in so doing, they cause a pyogenic immune response. Now, one of the problems with the innate immune response is that these phagocytes and these eosinophils, they're not very specific about what they damage. They swarm to the area, they go running to the area where the foreign invaders are, And once they're there, they just start attacking stuff and destroying the foreign invaders, but they also have the effect of damaging your own tissues as well. So here's an analogy that I'm going to use for the immune system, which you'll hear over and over again. The innate immune response, particularly the pyogenic immune response, is kind of similar to the riot police. Right? I don't know if you've ever seen riot police when they go into action. They come into an area where there's civil unrest, and they have truncheons and shields and helmets, and they come in there and they shoot tear gas around, and they treat everybody like they're a criminal. They say, everyone get out of here, or we're going to hit you on the head with our truncheon. We're going to arrest everyone that doesn't leave. They treat everyone like they're a criminal because it's a fairly aggressive and emergency response from the police, right? So that is essentially what the innate immune system does, and all of these granulocytes, that's kind of what they do. So what that means is that if you call out, if the riot police get called into an area, if you're an innocent civilian, if you're there, you're probably going to get arrested or hit on the head with a truncheon. So the riot police kind of accidentally many times attack and damage good people, right? So that's what happens when the innate immune response is brought into play. These the phagocytes and various other granulocytes come into the area, and they will kill the bad guys, but they will also damage some of the good tissue. Okay, that's not necessarily good, but the advantage is that it's fast, right? There are many times when you need a fast response, and who cares? The fact that it will damage your tissues as well as the bad guys is bad, but it's not as bad as letting the bad guys stay there. That's basically what's going on. Okay, now if you don't want that kind of an emergency, blunt, indiscriminate response to invaders, then you invoke the adaptive immune system. So in the adaptive immune system, you send in the nice, friendly neighborhood police who know what everyone in their neighborhood looks like, and they can recognize the criminals as being not meant to be there. But those nice neighborhood police, they may take a while to go and get their riot gear on or something before they arrest the bad guy. So the adaptive immune response is good because the adaptive immune response, where you produce antibodies to attack a foreign invader, the production of those antibodies will almost never damage your tissue. So the humoral immune response, the adaptive immune response, is a more specific attack on the foreign invader, where the body destroys the foreign invader, but it doesn't destroy any of your tissues. That's the good part. The bad part is that it takes about 2 weeks for your body to mount an adaptive immune response. And that, for instance, is why it takes you 2 weeks to get over a head cold, because a new virus comes in. Various other cells, something called an antigen presenting cell has to look at it, and then a T helper cell has to look at that, and then the T helper cell goes and finds some B cells that are capable of making antibodies that will attack and destroy that particular virus. That whole process is very complicated, and it takes about 2 weeks to do that, and that's why it takes you 2 weeks to get over a cold or a flu, at least. Okay, so the innate immune response is like the riot police. They destroy the bad guys quickly because they are born knowing what they look like, what certain bad guys look like. Not all of them, but certain bad guys like Staphylococcus aureus. Whereas the neighborhood police, the adaptive immune response, are going through your neighborhood looking for a specific criminal, and they know what that criminal looks like. So if you're not that criminal, you don't have to worry. However, the bad news is that it takes about 2 weeks. It's a fairly slow response. All right, there are other types of cells that I haven't mentioned yet. There is a type of cell called a natural killer cell. We're not going to discuss that in this course. Phagocytes are given different names. Their job is to eat things, and they usually recognize who it is they're supposed to eat from the beginning. So this is part of the innate immune response. And then we give them different names depending on where they are in the body. So monocytes, neutrophils, and macrophages are all examples of a class of immune system white blood cell called phagocytes. Mast cells and basophils are involved in the inflammatory response. Eosinophils are specifically there to destroy microscopic worms that have gotten into your body. The word eos is an ancient Greek word meaning pink. Right, so now we know some ancient Greek words. We know that erythro was the ancient Greek word for red. We know that leuko was the original ancient Greek word for white. We know that eos is the original Greek word for pink, and by the way, the original ancient Greek word for yellow is xantho. Right, so if sometimes there are various conditions where somebody will have yellow spots on their skin, you call those xanthoma, if you have yellow spots here and there. All right, so then these things here are all part of the innate immune response. Then we have the T cells that basically tell the B cells what to do. It goes and finds certain specific B cells and tells them to differentiate into plasma cells and then start producing antibodies that will kill a certain specific bad guy. Okay, so these cells all belong to the myeloid lineage, and they are all granulocytes. These cells all belong to the lymphoid lineage. These cells are all part of the innate immune response, the innate immune system. These cells are part of the adaptive immune system. These cells are all part of what is referred to as the cell-mediated immune system. These cells are part of what is referred to as the humoral immune system. So you need to be familiar with all of these cells and what type of immune response they mediate. Which cells are examples of ones that do there are granulocytes, which ones are non-granulocytes, which ones we talked in the blood cell section about PMN cells, polymorphonuclear cells. You have to know which ones. Those are generally granulocytes that are members of the myeloid lineage. It may seem overwhelming to learn the names of all these cells, but if you take an immunology course, you will have to go into this in even greater detail. Believe it or not, you spend a whole course, a whole semester, just getting a so-called introduction to all these cells and what they do. Okay, so the granulocytes are members of the myeloid lineage, and we know already that they have a polymorphic nuclei. In fact, none of these cells have nice round nuclei, but whether they have 2 lobes or 3 lobes is specific to the cell type. If you're going to be an immunologist or a hematologist, you have to know how to recognize which type of granulocyte has which shape of nucleus. So that's something, but we're not going to learn that in this course. And the lymphocytes, B cells and T cells is all we're going to worry about this time. All right, so you can see here that we have a bunch of erythrocytes in this slide, and then we have 2 different types of polymorphonuclear cells. These are 2 different granulocytes. And here's a nice, lovely lymphocyte. You can tell a granulocyte from a lymphocyte mainly because the granulocytes have a more grainy appearance, but the lymphocyte, and by the way, this lymphocyte looks pink, but it's not an eosinophil, by the way. It looks pink just because of the way the slide is stained. But you can tell the lymphocyte because it is a very lovely, lovely round nucleus that takes up most of the cytoplasm. So as we said, phagocytes eat invaders. Now, phagocytes, once they've eaten a foreign bacteria, for instance, they later become a cell that is, they're renamed an antigen-presenting cell, which is a key step in the humoral immune response, which we're going to talk about next. Right, so remember when we get to the humoral immune response and I talk about APCs, antigen-presenting cells, I am talking about phagocytes, one of the phagocytes that has eaten a foreign invader and then presented that foreign invader's antigens on its own surface, and it presents those foreign antigens to a T helper cell. So we'll talk about that later. So these are some of the locations where you would find different types of phagocytes. Right? So you would find neutrophils, they are a phagocyte. You would find monocytes in the blood, for instance, they're a phagocyte. You would find macrophages, which are also a phagocyte. You'd find them inside the lymph nodes. You would find a type of phagocyte called a dendritic cell inside the skin. So here we have a nice image of a phagocyte engulfing a cancer cell and destroying it to prevent somebody from getting cancer. Here we have another beautiful picture of a phagocyte spearing and eating bacteria. So what's happening here is that these cells are these little cells are bacteria These little cells are bacteria, and they are being speared by these kind of spears that the phagocyte is sending out called pseudopods. And then the pseudopods, kind of like an octopus, reaches out and grabs something and then brings it back and puts it into its mouth. Well, the phagocyte doesn't have a mouth, but it engulfs things through phagocytosis. And in order to do that, you have to be attached to the skin or the surface, right? So it reaches out and grabs these things and then puts them on the skin or the plasma membrane of the cell, and then they get engulfed and taken inside. As I said, the pyogenic response is where you have pus formed because certain phagocytes recognize certain types of bad guys on site. Pyogenic bacteria are bacteria that are classified as pyogenic bacteria because they provoke the formation of pus. Examples of pyogenic bacteria are Staphylococcus aureus and Streptococcus pyogenes. In fact, Streptococcus pyogenes has the word pyogenes or pyogenic in the name. Okay, so inflammation is where mast cells secrete histamine, causing swelling and inflammation. If you have an allergy, that's referred to as a type 1 hypersensitivity reaction. That is where mast cells dump out histamine and give you, if you inhale some pollen, the pollen sets off something called a type 1 hypersensitivity reaction if you have an allergy, right? So here's a mast cell which is doing something called degraining. A pollen grain or some other type of an antigen has made contact with the surface of the mast cell, and as a result of that, these granules inside the cytoplasm of the mast cell have dumped out all of the histamine that they were containing all over the place. That causes the soft tissues around your eyes and your nose to swell up and become red and become irritated, and you get hay fever, right? So you can see in this person, you can see soft tissues around, all the soft tissues around the eyes have swollen up due to the leakage from the capillaries. And of course, if you want to avoid that, you take antihistamine tablets, which is a chemical that makes it difficult for the mast cells to degranulate. Eosinophils are specialized to destroy worms by dumping enzymes on them. Here's an eosinophil. It looks very pink under the microscope, mainly because of the way we stained it, but that's an eosinophil. So here we have a microscopic worm. If you take Biology 205, you'll learn that the name for parasitic microscopic worms is helminths, right? So there's a whole class of invading microbes that are called helminths that will get inside and make you sick. And here we have eosinophils that are dumping proteases directly onto these microscopic worms to kill them. Right, now I'm going to show you a slightly disturbing image here about what microscopic worms, specific types of microscopic worms can do to you. So get ready, brace yourself for a bit of a shock. Okay, this isn't it. This is simply a microscope picture of eosinophils attacking a worm called Wuchereria bancrofti. Wuchereria bancrofti causes a disease called, brace yourself, called elephantiasis. So this poor woman had elephantiasis in the 19th century. Elephantiasis is caused by a microscopic worm called Wuchereria bancrofti, which is carried by mosquitoes. Mosquitoes are generally a nuisance to people and carry a lot of different diseases, including elephantiasis. So if you're bitten by a mosquito that's carrying Wuchereria bancrofti, these worms are exactly the right size needed to block your lymphatic vessels. And if you block your lymphatic vessels, they cannot drain the surplus excess fluid out of the interstitial spaces, and you get massive amounts of swelling. If it happens to one of your limbs, like the arms or the legs, the limb will end up looking like a big elephant trunk. It'll swell up and turn gray and be folded like an elephant trunk, and so they call it elephantiasis. Trichinosis is another type of helminth disease that you can get if you eat meat from animals that you've hunted. So if you go out and you hunt moose or you hunt animals from the forest, you shoot them and then you cook them, you have to make sure that you cook them very well because they often contain a microscopic worm called Trichinella spiralis. That worm is living. That worm drills its way into the muscles of the animal that you shot, and then when you eat it, they drill their way into your muscles, and then you get the same disease the animal had, which is called trichiniasis or trichomoniasis. They've changed the name recently, so trichinosis is the name that I remember. Right, so if you eat meat that you've hunted, you could get trichinosis. Also, if you eat meat from an unregulated animal farm, so you have a pig farm where there's some guy that's just raising a few of his own pigs. They haven't been inspected by federal health inspectors, and so the pigs could be carrying trichiniasis or other parasites. Tinea solea is what's known as a tapeworm, so pigs often carry that. Pork tapeworm, it's called. So you get this stupid little worm that's in your food if you didn't cook the pork well enough, and then this worm, the eggs from this worm get into your intestine. They latch onto the inside of your intestine, and they just keep on growing because they're stealing your food as you're digesting it, and they keep growing. And it's not uncommon for Tinea soleum, to this tapeworm, to be 10 meters long. So you have a large 10-meter long worm in your intestine, all because you ate some pork that came from a farm that wasn't inspected properly. Okay, so just to drive this point home, the innate immune response is pre-programmed and takes no time at all. It involves phagocytosis, for instance. The pyogenic response is part of the innate immune response. And it's characterized by the formation of pus. These are the cells that carry it out, phagocytes such as monocytes, neutrophils, macrophages, and dendritic cells. It is nonspecific, so it will also damage your own cells in the area, kind of like the riot police. The mast cell produces the inflammatory response. Eosinophils are responsible for directly attacking microscopic worms with proteases. And there are two types of immune responses: the innate cell-mediated or pyogenic inflammation response, which is mediated by myeloid and members of the myeloid lineage that are granulocytes, versus the humoral or adoptive immune response. Let's talk for a minute about inflammation some more. So during inflammation, mast cells release histamine and other things as a response to something called an insult, a physical insult. An insult can be pressure, like if you bang your hand on something, it will turn red and swell up, and that's referred to as an insult. Anything that does moderate damage to a body part is referred to sometimes as an insult. And or if you have an allergy, you have inflammation of the tissues around your nose and your eyes. Usually it's in response to, in the case of allergy, it's in response to something called a superantigen, which we won't talk about now. But basically, as you know, an antigen is something that provokes the formation of antibodies. A superantigen is something that skips a whole bunch of steps and leads directly to inflammation rather than the regular humoral response. Okay, so what does inflammation look like? Remember that inflammation, and you're usually asked this on a final exam, what are the four characteristics of inflammation? And the four characteristics are redness, pain, swelling, and a sensation of heat. Those are the four things that characterize inflammation. So here we have some examples of that. Right, so if you hit somebody, if you have hay fever up on the top left, you hit somebody, or you have hay fever. Right, you look like this because your eyes are your eyes are red and your nose is red because the capillaries are leaky in the soft tissue around your eyes and your nose, and so they're leaking out erythrocytes, red blood cells, into the area. Many people have allergic reactions to insect bites because their immune system hates insect bites, right? And so then you have the same inflammation there. Many people, the inflammatory response is also mediated by venom. Wasp venom, for instance, is a special type of venom. If you look at a wasp stinger, it's actually very small. If you got poked with a pin, it wouldn't hurt nearly as much as if you get stung by a wasp stinger, which is much smaller than a pin. So how do you account for the fact that the wasp sting stings so badly? And it's because the venom that they carry actually activates pain neurons in the area. So the wasp is injecting you with this little tiny stinger, and then it shoots a bunch of this liquid into you that makes your pain receptors go crazy and hurt. but that generally causes an inflammatory response. The inflammatory response is actually an allergic reaction. Slightly, it's an immune system reaction to the venom. And finally, if you have a physical insult, like a punch to the eye, you end up getting the same inflammatory response. So, as I said, this is kind of like the riot police. They come into the area and they start damaging criminals, of course, but they also damage regular people who are not criminals. If you have chronic inflammation, this leads to fibrosis and scarring tissue. Okay, now let's take a closer look at the humoral immune response. What you do first is this refers to attack of foreign invaders by antibodies that are produced by plasma cells. Plasma cells used to be B cells. Right? And antibodies are very specific. Antibodies will bind or attach to only a specific foreign antigen. They will not attach to any other tissues, and so they will attach to the foreign invader. These antibody attacks are coordinated by a type of lymphocyte called a T helper cell. They learn how to recognize your MHC class proteins from foreign MHC class proteins, so they never go and get a B cell that will attack your proteins. They go and get a B cell that will attack only foreign proteins, which they know how to recognize. This response takes about 2 weeks, which is the bad part. Other than that, it's actually better than the innate immune response. All right, now this is going to be very difficult to follow. Most people find this extremely confusing. I wouldn't worry about it too much because very little of the final exam is going to be on the humoral adaptive immune response, of how the sequence of events that leads to the production of antibodies that will destroy a specific invader. We're not going to dwell on that too much on the final exam. But try to understand it, because if you go on in the field of immunology or hematology, you'll have to know this. All right, an invader comes into the body. That's step one, and it gets eaten by a phagocyte. Right, so the phagocyte, even though the phagocyte may not recognize this thing as being foreign, one or two phagocytes will still eat it, like a macrophage or something like that. This phagocyte, one of the things that it does is it takes the foreign invader apart and literally puts some of its foreign antigens onto its own surface. And then it shows the foreign antigen to a T helper cell. Right, so the analogy that I like to use is, imagine you had a police dog, right? So the policeman finds a piece of a bad guy's shirt, and he takes it to the dog trainer, takes the piece of the shirt to the dog and lets the dog sniff it and says, "Go find the bad guy." Right? So the dog will run and find the bad guy and kill him. That's basically, in this case, in this analogy, the T helper cell and the antigen presenting cell are like the dog trainer. The antigen presenting cell is like the dog trainer, and then the T helper cell is kind of like the dog. Right? So the dog is not going to take this order from anybody. He'll only take it from his trainer. Right? So the trainer takes a piece of the bad guy's shirt, presents it to the dog, and says, "You see what that smells like? Okay, go find the bad guy." So in this case, a phagocyte engulfs or eats a foreign bacteria. It takes it apart and then puts some of the foreign antigens onto its own surface. A T helper cell then comes and makes contact with the antigen presenting cell, as it's now called. And then it recognizes that this is one of its own phagocytes, presenting it with the antigen that is found on the surface of the bad guy. So then the T helper cell then goes and finds a B cell or a series of B cells that will make antibodies that will bind specifically to that foreign antigen. Okay, so here we have a phagocyte eating a bacteria, right? And then it takes it apart, and these foreign antigens that you're finding on the surface of the bacteria cell get taken into the phagocyte, and then it puts them onto its own surface and then presents them to a T helper cell. The T helper cell then goes and finds a B cell that can make antibodies that can stick to that particular antigen, foreign antigen. Okay, here's something that very few people know. The immune system has a pre-existing repertoire, kind of like a library of B cells that are able to make antibodies to specific foreign antigens. But we only have about 2.5 or 3 million different types of B cells that are capable of making around 2.5 or 3 million different types of antibodies that will bind to 2.5 or 3 million different foreign antigens. That's a lot, but we don't have an unlimited amount. But nevertheless, the T helper cell then basically, this is a simplified explanation, but the T helper cell then goes and finds a B cell that can make an antibody that can bind to that particular antigen that it was just presented with, but from the antigen presenting cell. And then it it selects that B cell that's capable of making that antibody. And that is called clonal selection. Clonal selection. The T helper cell then produces cytokines that cause that B cell to multiply. So now instead of having 1 or 2, we have 1 or 2 million like that. That's referred to as clonal expansion. Right? And then now we've suddenly got an army of these B cells that can make an antibody that can attack that foreign bacteria. And they are made, they differentiate into plasma cells who release their antibodies into the blood plasma and into the interstitial fluid and attack and destroy that invader. All right, so we have a T helper cell has just been presented with a foreign antigen from the antigen presenting cell. It then goes and finds a B cell that can make an antibody that will bind to that particular foreign antigen. That's called clonal selection. It stimulates that B cell to divide and multiply or proliferate. That's called clonal expansion. And now we have a whole army of that B cell that can make antibodies that can destroy that foreign invader. And these B cells differentiate into a type of cell called a plasma cell, which is specially designed to release these antibodies. Right, so the T helper cell goes and finds a B cell that can make an antibody that will bind to this particular foreign antigen. It causes them to multiply and differentiate into plasma cells. Right? And then we have an army of these things that are sending out these antibodies into the blood and the plasma and the interstitial fluid to bind to the foreign invader. And we call them plasma cells. And once the foreign invader is destroyed, here's something interesting. We have a subset of those B cells that kind of took off on their own, and instead of turning into plasma cells, they turned into something called memory B cells. Memory cells. And those guys are left behind after the battle is over in case that bad guy dares to show up again. They're ready for it. Right? So these memory cells are left behind after we've killed the foreign invader. These are left behind in case that bacteria comes back, and they're ready for it immediately. We don't have to go through all of this again because we left behind a whole bunch of what are called memory cells or memory B cells that are ready for it next time. Okay, that, by the way, is the principle behind vaccination. This is where you take a bacteria, for instance, or a virus, and you grow it up and you kill it, and then you inject it into somebody, and their immune system will mount an immune response based on the fact that that bacteria is made out of foreign proteins. And the bacteria can't hurt you because it's dead, but your immune system can still go and find some B cells that can make antibodies, and then so that your immune system will mount an immune response to the dead bacteria, but it's dead. You don't really care about that part. But at the end of all of that, it leaves behind all of these memory cells that are ready for it next time. And so you've trained your immune system to destroy that bacteria the next time it sees it. And the next time you see it, it'll be alive. This is what a plasma cell looks like. What is all this stuff? Right? We talked at the very beginning of the course about cell organelles. This is endoplasmic reticulum that is intended to build proteins that are meant for export. So antibodies are made of protein, and these plasma cells are secreting massive amounts of antibodies, and so they need a lot of endoplasmic reticulum. So that's what all of this is. All right. So how do antibodies go about destroying a foreign invader? Okay, so it's not quite that simple. The antibodies themselves don't actually destroy the foreign invader. The antibodies merely attach themselves to the foreign invader's proteins. Okay, so how does that end up destroying the foreign invader? Okay, so antibodies will attach themselves to the proteins on the foreign bacteria or the foreign virus. When you have antibodies attached to the outer surface, that is referred to as opsonization. So you do need to know for the final exam what opsonization is. Opsonization makes it easier for phagocytes to eat the invaders, right? So that's one way that antibodies help to destroy the foreign invader. So remember, antibodies by themselves do not kill the foreign invader, they merely attach to it. So how does the attachment of these antibodies lead to killing the foreign invader? So attachment of the antibodies is called opsonization. Once a bacteria or a virus has been opsonized, it is much easier for phagocytes to eat it. Right? So the bacteria or the virus that has been optimized is often eaten by a whole bunch of phagocytes that now find it easier to eat the virus or the bacteria because it's covered by antibodies. Okay, but we don't stop there. There are a bunch of proteins in the serum called complement proteins. Those complement proteins are produced by the liver again, right? So there's another awesome thing that the liver does. Remember, on the final exam, I might ask you to list 10 things that the liver does. And these complement proteins bind to the antibodies, right? And these complement proteins punch holes in the plasma membrane of the virus or the bacteria. Right, so two ways that opsonization kills a bacteria or a virus. One is it makes it easier for phagocytes to eat it. Another way is that the antibodies will, proteins called complement proteins will bind to the antibodies and then punch holes in the plasma membrane of the bacteria or the virus. And then finally, after we've killed all the invaders, we leave behind memory T cells and things called memory B cells that can respond quickly if that same invading bacteria or virus ever shows up again. And the production, leaving those memory B cells and memory T cells behind, are the basis behind vaccinations. So here we see opsonization by antibodies, followed by phagocytosis by phagocytes. And over here we see antibodies opsonization, so the antibodies are stuck to the surface, and then these complement proteins that are produced by the liver bind to the antibodies and then punch holes, pores in the plasma membrane of the cell and destroy it that way. We leave behind memory cells. So the summary of the response, the humoral response or the adaptive response, is that the invader is first eaten by a phagocyte. It doesn't happen very often, but once or twice is enough. It then takes it apart and presents the foreign antigens from that invader on its own surface. It therefore becomes an antigen presenting cell. Who does it present the antigens to? It presents it to helper T cells. The helper T cell reads the signal, and then the signal being the fact that, this is a cell that has my human HLA complex on it, but it's presenting me with foreign antigens as well. I'm a police dog. This is my trainer presenting the bad guy's shirt to me. Now I'm going to go off and find him. So the T helper cell, the police dog, then runs off and gets some B cells and then causes them to expand, clonal selection and clonal expansion. Those B cells differentiate into plasma cells, which secrete antibodies which opsonize the foreign invader, and the foreign invader is then destroyed by more phagocytes or by complement proteins. And at the end, we leave memory cells behind. This process takes about 2 weeks. And notice that the T helper cells are the weak link in the chain. Right? So we're going to talk in a minute about the human immunodeficiency disease, HIV, human immunodeficiency syndrome, the AIDS syndrome. The AIDS syndrome is caused by a virus called the human immunodeficiency virus, HIV. The human immunodeficiency virus invades and destroys T helper cells, and thus causes the coordination of the immune system to fall apart. Right? So it causes destruction of, essentially, if you think of the If you think of a T helper cell as a general, if you kill the general, then the soldiers don't know what to do anymore. That's a good, very effective way of destroying the immune system. So the T helper cells are the target of the human immunodeficiency virus, the HIV/AIDS virus. As I said, the T helper cells are educated or selected in the thymus. That's why they're called. That's where the T in T helper cell comes from. It refers to the thymus, as opposed to B cells. The B stands simply for bone marrow. So both the T cells and the B cells are born in the bone marrow, but the B cells simply then go out into the blood and do whatever B cells do, and the T cells have to go through the thymus before they're allowed to work. So this diagram shows the sequence of events, basically. Okay, so first the foreign invader is eaten by a phagocyte, which turns into an antigen-presenting cell. The antigen-presenting cell presents the foreign antigens to a T helper cell. The T helper cell then goes and finds B cells that are capable of making antibodies that can destroy that foreign antigen through opsonization and so on. It also goes and finds some cells that are called cytotoxic T cells, which we're not really going to deal with in this course. And then after the invasion is over, the war is over, we leave behind some memory T cells and some memory B cells and some memory cytotoxic T cells that are ready for the invader if it ever comes back. And that's the principle behind vaccination. All right. Now, so I mentioned that the adaptive immune response takes about 2 weeks. When that happens, that's referred to as a primary immune response. After that response is over with, we leave behind the memory cells so that we're ready for it next time. The second time we see it, the immune response is immediate because we've already got the B cells ready to go. And that is referred to as a secondary immune response. And it is much stronger and much faster. The first time your immune system sees this foreign invader, it takes about 2 weeks for it to round up all of these B cells and make them differentiate into plasma cells. The next time you see it, your immune system is ready for it immediately. It responds immediately, and the number of antibodies that's produced is more than 1,000 times greater than the first time you saw it. So it's a very fast, very strong immune response the next time you see it. Vaccination is where you inject somebody with a dead virus or a dead bacteria. A virus is not really a living thing to begin with, but I'm just using an analogy. So with a vaccination, you grow up a bunch of viruses or you grow up a bunch of bacteria and then you kill them and then you inject them into somebody. Their immune system will mount the primary immune response anyway, even though the bacteria or the virus can't hurt you because it's dead. But then your immune system will leave behind the memory cells and you'll be ready for it the next time you see it, and the next time you see it, it will be alive. And your immune system is actually able to kill it before it has time to invade your cells. The word vaca means cow, by the way. It's a Latin word for cow, and it started out with a scientist named Edward Jenner who discovered the first vaccination to smallpox, which is a terrible disease which has now been eradicated or eliminated. But what happened was that there was a disease called cowpox, and Edward Jenner in the 18th century knew that if you caught cowpox, you were then immune from smallpox. And so he took pus from the pustules, from the blisters of people who had cowpox, and literally injected them into people who didn't have cowpox, and then found out that that prevented the people that you injected from getting smallpox, and so that was referred to as he called that a vaccine after the cowpox. But nowadays it means just what I just explained. You grow up a bunch of viruses or bacteria, you kill them, and then you inject the dead viruses into a person. Your immune system will mount an immune response to the dead antigens, and then it will mount the secondary immune response the first time you see it for real. So you grow up a culture of bacteria or viruses. Viruses are hard to grow because they're not living things. They won't live on their own, so you have to grow them inside some type of cells. A very common way that you grow the flu vaccine, the viruses that cause the flu, the influenza virus, is to grow them inside the eggs of chickens, because most influenza viruses live in birds as well as humans. And so how do you grow viruses that are not living things? They have to grow inside live cells. One of the ways you do that is by growing them up inside fertilized chicken eggs. Okay, then you kill the viruses and you have a vaccine. You destroy them either by heating them up or by mixing them with a chemical called formalin. And then you inject the dead organisms into a person. You vaccinate them. And then the organism can't hurt you because it's dead, but you mount a primary humoral response anyway. And then you leave memory cells behind. Okay, so this graph is showing the number of days that you were exposed to a virus, for instance, on the x-axis, and how many antibodies is your immune system producing on the y-axis. All right, so let's say we do this experiment. You inject somebody with a live virus or a dead virus on day 1, and then we look at how long it takes for their immune system to start making antibodies. So at first, a week later, your system starts to make a few antibodies, not that many. Then we get more and more and more. And then after about 2 weeks, your immune system, after 14 days or 2 weeks, your immune system will kill the virus. And then there's no need to make all those antibodies. So the production of antibodies drops, but we left behind memory cells. So on day 28, we inject somebody with the same virus again, and bang, look at the response of antibody production. The production of antibodies is immediate, and look at the scale of the x-axis. Right? So here we have 10 to the first antibodies per mil here, and then during the secondary immune response, we have nearly 10 to the fourth. Right? So the production of antibodies is immediate, the same day, and we start producing nearly 1,000 times more antibodies than we did the first time. So much so that you can destroy, your body will destroy, your immune system will destroy the virus or the bacteria before it has a chance to infect you. That's the principle behind vaccination. Now over here we have what's called an experimental control. This is where somebody injected a different virus on day 35 to see if, is it the second time you see a virus in general that you get this response? No. It is specifically the second time you see that specific virus. If you inject another virus, you go through the primary immune response the first time. I mentioned the fact that bacteria can grow fine in a bottle. If you give them some sugary water, you can grow bacteria quite easily. But viruses are not living things. Viruses are kind of rogue genes that invade a foreign cell and then usurp or hijack the metabolic machinery in that cell to make more viruses. So how do you grow them up? Well, there are a couple of ways you can do it. You can grow them in chicken eggs, as I mentioned. That's usually what they do in the case of the flu. And that's one of the reasons why when you get a flu vaccination, they ask you if you are allergic to eggs, because they've been growing the vaccine viruses inside eggs. Or some human cells you can grow inside a bottle. Those are called cultured human cells. If you take Biology 200, we'll tell you all about what human cultured cells are, but basically you're just growing human cells in a bottle. So you can infect those human cells with a virus as well. Or thirdly, you can grow them up inside certain types of animals. They used to grow certain vaccines inside pigs or horses sometimes. They inject a live bacteria inside a horse. And then the horse actually makes antibodies in its own blood, and then you take the antibodies out of the horse's blood and you inject them into a person who's been infected with something. Those viruses will attack and destroy the virus or the whatever it is that infected you, but you didn't actually make the antibodies yourself. You simply stole them from a horse that made them for you. When you do that, that's referred to as a passive vaccine. That's where you produce antibodies in another animal and then you inject them into you in order to cure you of the disease, but it was actually somebody else, another animal, that made the antibodies. Okay, here's a factory that's making influenza virus en masse, huge quantities of influenza virus by injecting chicken eggs. how do you kill the vaccine, how do you kill the bacteria or the virus before you inject it? One of the ways you can kill it is by heating it up. That usually denatures or destroys some of the proteins. That's what they did with the original polio vaccine. Polio is a terrible disease that if you get it, it's caused by a virus, caused by the polio virus. If you get a bad case of the polio virus, it can actually paralyze you completely. If you get a mild case of it, you may just end up being paralyzed from the waist down, for instance. Might end up being a paraplegic instead of a quadriplegic. Another way you can get rid of them is by chemical inactivation. So you mix them with formalin, or which is a derivative of formaldehyde. There was that was another way that they made the original polio vaccine. Okay, there is a thing called a recombinant vaccine, which is where you don't actually inject somebody with the whole virus or the whole bacteria. You simply make the protein another way, and you inject them with the viral or the bacterial protein. That's a nicer way to do it because the virus or the bacteria was never alive, so you don't have to worry about the fact that when you made the vaccine you might not have killed all of them. You just injected the proteins by themselves, right? But that happens to be a very expensive way to do it. It's not done very often. All right, here we have two different versions of the polio vaccine. So as I said, polio causes paralysis of the nerves. There was a famous scientist named Jonas Salk who invented the heat inactivated polio virus in the 50s. And a lot of people who were born in 1950 or thereabouts were injected with the heat inactivated Salk vaccine. Then there was another scientist named Albert Sabin who invented an interesting type of a vaccine that was referred to as a live attenuated virus. So he took the polio virus and mutated it so that it was harmless. And because polio virus gets into your body through what's known as the fecal oral route, you get it by swallowing water that's contaminated with sewage. The polio virus gets into your body through the digestive tract, right? So with the Sabin live attenuated vaccine, you could simply swallow it, right? And so people like that better because they didn't have to get a needle, right? But it turned out that some of those, well, first of all, the Sabin virus, the live attenuated virus, was invented in the first place because when Jonas Salk took the polio viruses and heat inactivated them, they didn't succeed in inactivating 100% of them. So they injected millions of people with the polio virus, and 1 or 2 people actually developed polio from the vaccine because you didn't kill 100% of the viruses when you made it. You killed 99.99999%, but 1 or 2 people actually contracted polio from the vaccine. And there was a big legal fuss and lawsuits and all kinds of stuff happened, which I'm not going to go into. But that prompted Albert Sabin to make a mutant version of the polio virus, which is harmless. And then people started taking that instead. And then somewhere around 1970 or so, or 60 or so, something like that, some people ended up getting polio from the Sabin vaccine as well because if you know anything about genetics, you know that if you have a mutation that changes the DNA sequence and changes the proteins on the surface or whatever, you can have something called a back mutation where the virus spontaneously mutates back to the harmful form. So people were getting sued over the Sabin vaccine as well, and they went back to the Salk vaccine. Okay, so I was injected with the Salk vaccine. Many people were. I was injected at my school in 1968, actually. No, I'm sorry. In 1968. I was given the oral version, right? So I was given the oral version. I did not get an injection. And as a 6-year-old kid, I was very relieved that I didn't get an injection. But I could have gotten polio. I didn't realize that, of course. Okay, now the question remains, why can't we make a vaccine to every bacteria or every virus? Remember that you have a pre-existing repertoire of 2.5 million types of B cells that can make antibodies that bind to 2.5 million different foreign antigens, but we don't have an unlimited number of B cells that can respond to an unlimited number of foreign proteins. And so sometimes if we have good luck, whatever virus is plaguing the human race, whatever virus is plaguing us, we happen to have an antibody that can kill it, And so if you kill that virus and inject it, then the if you kill that virus and inject it, then the antibodies will go to work and leave behind memory cells. On the other hand, if you have a virus that we don't happen to have a B cell that can make antibodies against it, then we're out of luck. Right? So, all right. So we've had some success and some failures when it comes to making vaccines, depending entirely on whether or not we have a B cell that's capable of making antibodies. Okay, so for example, we've had great success with polio. Polio has been eradicated. Smallpox has been eradicated. These were once horrible diseases that made people's lives terribly miserable. We have eradicated or controlled rubella, which is a very contagious disease that used to, it would kill a few children every year, but more commonly it would cause a few children to go blind or deaf until we invented the rubella vaccine. Chickenpox as well. There is a vaccine for HPV-16, which causes cervical cancer in women. So we've been able to make successful vaccines against all of these things. Right? As an example, polio. This is a 1930s iron lung wing of a hospital. You see people laying on their backs inside these machines, which was called an iron lung. These iron lungs were all filled with children, and these children were unfortunate children who caught the worst possible cases of polio, and they were paralyzed from the neck down and they were unable to breathe. And this is back in the days before we invented antibiotics, and so we couldn't do tracheotomies where you put you have a ventilator that goes directly into your lungs. The only way they could get these children to survive by breathing was to put them in an iron lung. So an iron lung was a machine that was attached to a vacuum pump that would lower the pressure inside the drum, and that would cause the lungs to expand, and then they would release the pressure and they would breathe out. Right? So this was a terrible disease that you had thousands of these poor, unfortunate children spending their entire lives in iron lungs. So getting rid of polio was an extremely good thing. Today, there are people, despite having very good success stories in making vaccines that got eradicated diseases that plagued the human race and made life very miserable for many people, there are people who are now, for various reasons, reasons that they find out about on the internet and so on, that are against vaccinations. For example, the measles, mumps, rubella vaccine, also known as the measles, mumps, rubella, varicella zoster vaccine, is given to children when they're 13 months old, and it vaccinates you against the measles, the mumps, and the German measles, which is known as rubella. These are very contagious diseases that either used to kill children or make them blind or deaf. They weren't terribly lethal. The mortality rate for these viruses was small, relatively small, but they were extremely contagious. Now, about 20 years ago, a doctor named Andrew Wakefield published a paper where he used a small number of children and compared 500 children who got the MMR vaccine to 500 children who didn't, and then claimed that there was a slightly higher number of children in the group that got the vaccination who also had autism or autism spectral disorder, which kick-started the idea on the internet and other popular media that the MMR vaccine therefore must be causing autism. Actually, nothing could be further from the truth. By the way, this is one of the things that the rubella vaccine prevents against is something called congenital rubella. So if a pregnant woman contracts rubella because she wasn't vaccinated against rubella, her child will be born with microcephaly and retardation. Microcephaly means a small head. Also born with heart problems and cataracts, vision problems. Okay, so the consequences of not being vaccinated against rubella can be fairly severe. But we have this movement now called the anti-vaccine movement, where people don't want their children to be vaccinated. But I wanted to show you a paper here. This paper that you can see for yourself, you can see the references, you can look it up for yourself. This paper was published in the New England Journal of Medicine, a very prestigious medical journal, in 2002, and it was a population-based study of measles, mumps, rubella vaccination and autism. Now that guy that I showed you before, Andrew Wakefield, published a small study that he did with about 1,000 children, and he allegedly showed that there is a slightly higher incidence of autism in the group that got the vaccine versus the group that did not. But later on, and that's the study everyone remembers, but very few people have heard of this study, and they should. So you as biologists should be telling everyone that will listen about this particular journal article. It was published in the New England Journal of Medicine. And it was done in Denmark. Now in Denmark, all of the medical records for everybody in Denmark is put into a central computer. The names and addresses and the identification information is removed, but how old are all these patients? What do they have? What did they get? How are they treated? All of that information is put into a central computer that researchers, epidemiology researchers, can study and look for patterns. Okay, so what they did here was this group in Denmark looked at roughly 500,000 children who were born in Denmark between 1990 and 2000, the year 2000. So they looked at roughly 500,000 children over a 10-year period. Of those 500,000 children, roughly 300,000 of them got the MMR vaccine, and 200,000 approximately did not get the vaccine. And then they looked at the percentage of the children in each of those two groups who developed autism, and they were identical. In fact, the percentage of children who developed autism in the group of children that got the vaccine was slightly lower than the children who did not get the vaccine. Right. So in science, we admit that you can never prove a negative. Right. So is it possible to prove that the MMR vaccine does not cause autism? No, because you cannot prove a negative. Logically, you can't prove a negative. But this is as close as you can get to absolute proof that the MMR vaccine does not cause autism, is not linked to autism. All right. What about other vaccines? Okay, so HPV-16 is a sexually transmitted disease. It causes a sexually transmitted disease called genital warts, and it causes these ugly warts to appear on the genitals of both men and women. But unfortunately for women, it also causes cervical cancer. There is now we have successfully created an HPV-16 vaccine, which will prevent women from getting genital warts and thus prevent women from getting cervical cancer. Right? So we've had a successful vaccine that will do away with that. Interestingly, people thought, well, that's an easy one. Why don't we vaccinate all of the girls before they become sexually active? Let's vaccinate them at age 12, 13, 14, 15. But a number of people thought, oh, we can't do that because HPV-16 genital warts is a sexually transmitted disease. If you vaccinate them to protect them from that disease, they will become promiscuous. Right? So we want to keep them scared and therefore celibate. We want to keep them scared enough that they won't have sex. So this is a political cartoon which is just showing that strange attitude. So here's a young woman who's dying of cervical cancer. It says HPV-induced cervical cancer on the chart, and the doctor is saying to the parents, I'm very sorry to tell you this, but your daughter has, and then the parents are yelling, sex! So it's a funny attitude, but it is an attitude. All right, we have never been able to make a vaccine against HIV. Another virus called the Epstein-Barr virus, which causes mononucleosis, which is a kissing disease. We've never been able to make a vaccine to destroy the herpes virus. And again, it's because we only have a pre-existing repertoire of B cells, and we just don't happen to have any B cells that can produce antibodies that will bind to the antigens on these viruses. So sometimes with vaccines, sometimes we have good luck and sometimes we have bad luck. All right, let's look at the lymphatic nodes themselves and see what's going on there, the lymphatic nodes and the lymphatic vessels. So we have a network of vessels in the body that are called the lymphatic vessels. They carry fluid called lymph, which was originally derived from the blood serum, drained out of the interstitial spaces. The lymphatic vessels have valves, right? So when we did the section on blood vessels, we said that arteries do not have valves, veins do have valves. lymphatic vessels also have valves, which should tell you that the lymphatic fluid is traveling rather slowly through the lymphatic system. Traveling through the lymphatic system at low pressure, therefore we need valves to make sure that the lymphatic fluid is flowing in the right direction. Okay, the network is punctuated by small organs called lymph nodes that are filled with white blood cells that are checking the lymphatic fluid for bad guys, bacteria and viruses. So they're kind of like police roadblocks that are on the road checking every car for criminals. You have a bunch of cars that are going through a police roadblock, and the police look inside to see if you have any bad bacteria. So the lymph nodes are filled with macrophages, B cells, and T cells. If they encounter a bad guy, those B cells and T cells will swell up, will multiply and swell up as they combat the bad guys, and that's why you get swollen lymph nodes. Ultimately, the lymphatic fluid is dumped back into the bloodstream at the level of the into the subclavian veins. Okay, so we dump the lymphatic fluid back into the bloodstream here. This is what a lymph node looks like. It's very small, and you can see that lymphatic vessels are going into the lymph node and going out through another route. Whenever you have something going in, it's referred to as using the word afferent, beginning with the letter A. So these are meant to be afferent lymphatic vessels, and then when something goes out, you use efferent, the word beginning with the letter E. So these are efferent lymphatic vessels. And these act sort of like police roadblocks, checking the lymphatic fluid to see if there are any bad guys in there. And if they do find any bad guys, they multiply and then kill them. Okay, now sometimes, unfortunately, when you get cancer, the cancer can either stay in the same place and form a tumor, or little cells can break off of the tumor and start flying around the bloodstream or the lymphatic stream. That is referred to as metastasis. This is kind of a funny pronunciation for this word, but the word meta means to not stay in the same place. Okay, so the word meta means to not stay in this, meta means above or beyond, I'm sorry. And stasis means to stand in the same place. So meta, to go beyond, stasis, standing in the same place. So metastasis or metastasis means that you have a cancerous tumor that the cells are flying off of the tumor off through the bloodstream or the lymphatic fluid. Those metastatic cancer cells can sometimes be trapped inside a lymph node and destroyed, and that's part of their job. So when you're sick, the doctor will check at least two sets of lymph nodes in your body, the axillary lymph nodes, which are often swollen up if you're having breast cancer, if you're fighting breast cancer, but other diseases as well. And the submandibular nodes, which are right underneath your jaw, which are often swollen if you're combating the flu or a viral disease called mumps. Right, so here we are checking the axillary lymph nodes, and here we are checking the submandibular lymph nodes. All right, now finally, as usual, we're going to talk about a little bit about the pathologies of the immune system, things that go wrong with it. An autoimmune disease is where you have improper selection or improper screening of the T cells. Two examples are rheumatoid arthritis and multiple sclerosis. And a third example is something called scarlet fever, which is where you make antibodies to a certain bacteria called Streptococcus pyogenes, and those antibodies cross-react, means that they cross-react with heart muscle, with myocardium. So cross-reaction means that you have an antibody that was made to bind to one protein, and you have another protein in your body which is unfortunately is very similar in shape to that protein, and so the antibody binds to that protein, your protein as well. So cross-reaction means that if you have antibodies that cross-react, it means that the antibody will actually stick to two different proteins that are similar enough that they look similar. And you made the antibody specifically to bind one protein, but unfortunately for you, it cross-reacts with another protein. So we'll talk about that. So those are examples of autoimmune diseases. There's also something called immune system evasion, which is how do you have some of these bad guys get into your bloodstream and they can actually avoid the immune system. They evade it. And we'll talk about 3 organisms that can do that. And then finally, the immune system destruction. The human immune system, the HIV virus, the AIDS syndrome virus, human immunodeficiency virus, as I said before, it invades and basically nullifies or kills T helper cells. The T helper cells are sort of like the general that's in command of the B cells. If you kill the general, the soldiers don't know what to do anymore, and that makes you vulnerable to a whole set of other diseases that your immune system would normally kill them, but because your immune system is suddenly uncoordinated and clueless, it can't. All right, autoimmune diseases, improper education of T cells. Rheumatoid arthritis is where antibodies attack the synovial fluid in the joints. Remember, we have synovial fluid in the joints. That synovial fluid contains proteins. Multiple sclerosis, we discussed this a little bit when we discussed the nervous system. Multiple sclerosis is where the antibodies that you have attack the myelin sheath of the neurons that travel down the spinal cord and also in the peripheral nervous system. Right. Rheumatoid arthritis, we have inflammation of the joints because we're having a sustained antibody attack on the proteins that are in the fluid, in the synovial fluid that cushions the joints. I showed you this actress before. She was very popular when I was a child. Her name's Annette Funicello. Very beautiful, successful woman. It was revealed when she was about 30 that she had multiple sclerosis, because when she was about 30, she started having to walk with a cane. Multiple sclerosis is where antibodies demyelinate your own neurons. By the time she was 40, she was in a wheelchair. By the time she was in her 50s, she'd lost the ability to move completely, and her husband even had to aspirate the saliva out of her mouth so she wouldn't choke. It's very tragic disease, all caused by demyelination of the neurons, antibodies cross-reacting with your own myelin. Okay, so scarlet fever is a disease that's caused by a bacterium called Staphylococcus. Is it staph or strep? Streptococcus pyogenes. Sorry, Streptococcus pyogenes has a protein in it called the M protein, a foreign antigen, and that protein is very similar in shape to your myocardium, to your heart muscle. So if you get an infection of Streptococcus pyogenes as a child, you will develop something called a strep throat. That strep throat sometimes leads to a generalized fever called scarlet fever. And your body will make antibodies that will bind to and attack the bacterial M protein, but the bacterial M protein is very similar to myocardium, and the antibodies cross-react with it, and you end up developing heart problems when you're in middle age. So many people die in middle age from heart problems as a result of having had scarlet fever when they were a child. These days, if your child gets a fever, hopefully you take the child to the doctor and you get antibiotics that will kill the Streptococcus pyogenes before your immune system starts mounting the primary immune response to it. This is the strawberry tongue, as it's called, a bright red tongue that you get when you have strep throat. And if you develop scarlet fever as a child, then you could have heart problems as a middle-aged person. All right, there are organisms called trypanosomes, which are an exotic kind of a tropical disease-causing organism that is spread by insects. And they are able to evade the immune system by doing something called antigen class switching. What that means is that they have funny proteins on their they have about 100 different genes that encode 100 different proteins that are on their surface, but they don't turn on all 100 of those genes at the same time. They turn on a few of them here this week, and then when they produce a new generation, the new generation turns on a different set. And what that means is that these trypanosomes are reproducing inside your blood, but they keep changing the proteins that are on their surface every 2 weeks, and thus staying ahead of your immune system. Right? So your immune system goes and mounts a primary immune response to the trypanosome the first time, and then it goes and gets some B cells and comes back 2 weeks later, and the trypanosome has changed the proteins on its surface, so the immune system doesn't recognize it anymore. Now there's a subgroup of there's a subgroup of trypanosomes that are called Leishmania tropica that cause a disease called leishmaniasis, which has some proteins on its surface that actually mollify or subdue the white blood cells that are there to eat it. Right, so that's an even worse thing, leishmaniasis. And we're not really sure how to deal with that, but it's an interesting, from a biology standpoint of view, it's very interesting. Okay, so these are trypanosomes. This is Trypanosoma cruzi. Trypanosoma cruzi is endemic to Central America, so southern Mexico, Nicaragua, Honduras, all those, Guatemala, all those places, and it is carried by an insect called a kissing bug. This ugly insect here. In Africa, the same thing is carried by an insect called a tsetse fly, and it causes an infection of these trypanosomes, which change the surface antigens on their surface every 2 weeks. So that means when your immune system is ready to kill and destroy the first variant of it, it disappears. And then 2 weeks later, you've got a slightly different proteins are appearing on the surface. And 2 weeks after that, you have another strain of the trypanosomes that look a little bit different. And every time, your immune system has to start over again every 2 weeks. And therefore, this particular protozoa stays ahead of your immune system. This is Leishmania tropica. There's another strain called Leishmania donovani. And what it does is it has proteins on its surface that actually tells the white blood cells to calm down and don't eat me, don't attack me. It's quite, we're not exactly sure how these proteins are able to do that, but it's fascinating. And so there's a lot of research going into it at the moment. You remember this scene from Star Wars where Obi-Wan Kenobi tells the stormtrooper, "Don't worry, these are not the droids that you're looking for." And then later on, that stormtrooper gets home that night and says, "But those were the droids I was looking for." That's exactly what Leishmania donovani does, or Leishmania tropicana does to the immune system. It turns off the white blood cells and tells the white blood cells, "Leave me alone. I know I'm not supposed to be here. I'm a foreign invader, but you don't need to bother with me." And so that's actually quite interesting. All right. Now there are some organisms, I mentioned at the very beginning that there's some that our immune system is very good at spotting foreign proteins, not very good at spotting foreign carbohydrates or foreign polysaccharides. Right? So there are some bacteria that get into your body and they clad themselves in a cloak of polysaccharides so that you can't see the proteins on their surface. So a glycocalyx is sometimes referred to as a slime layer or a slime layer, but the technical term is a glycocalyx. Example is Streptococcus pneumoniae, which is difficult to get rid of because your immune system has trouble recognizing it. So we have a regular bacteria here, right, that has no glycocalyx. And then here you can see this little halo around the strep pneumonia because this is all polysaccharide. Finally, the immune system destruction by HIV. This is where the human immunodeficiency virus invades and attacks and disables the T helper cells that coordinate the attack by B cells and plasma cells. And people who have the AIDS syndrome is a word to describe many symptoms and many characteristics. People who die from don't actually die from AIDS. They die because sadly they get infected by weird exotic diseases or weird forms of cancer that your immune system would normally take care of, so you wouldn't die from it. Okay, how do we kill bacteria besides the immune system? Well, one way we kill bacteria, this is a little side story about antibiotic abuse. We kill bacteria with antibiotics. And so antibiotics are drugs that kill bacteria. Antibiotics do not do anything to kill viruses. Right. So now there's an old saying that says anything that doesn't destroy you only makes you stronger, and that's not always true, but it is definitely true of bacteria. And that is when somebody is infected by a bacteria and you give them antibiotics to kill the bacteria, one or two of those bacteria will survive and be made stronger because there was something about them that made them resistant to the antibiotic that you used. And those, you will have knocked down the number of bacteria that are infecting you, right, for the moment, but then eventually the population of bacteria will be repopulated by this new strain of bacteria that is resistant to the antibiotic that you used. So as a result of that, the more often, as a race, the human race, the more often we use an antibiotic, the faster the bacteria that we're killing will catch up with us, because we'll kill most of the ones that weren't resistant, but we will have selected for the ones that are resistant. And so, for instance, the first antibiotic that was ever discovered was penicillin. It was discovered in the 30s. Now, almost all of the bacteria in the world, most all of the gram-positive bacteria in the world, are resistant to penicillin. And so, if you were infected by a gram-positive bacteria like Streptococcus pyogenes, the last thing in the world they would give you is penicillin, because it's almost guaranteed that the strain of Streptococcus pyogenes that's infecting you is resistant to it. Okay, so antibiotics kill bacteria, but not viruses at all. Examples of antibiotics are penicillin, methicillin, other things. If you expose a bacteria to an antibiotic, most of the time it'll die, but some will survive and be resistant. And as a race, if the human race uses antibiotics too often, eventually they will all be resistant, and antibiotics won't work anymore. Antibiotic abuse occurs when people who don't know that antibiotics will do nothing against colds and flus. Colds and flus are caused by viruses. Antibiotics do nothing to kill viruses; they only kill bacteria. The only way to prevent a cold or a flu is through vaccination. We have not yet succeeded in making any vaccines for colds because, well, I won't get into it, but colds are RNA viruses that change form fairly quickly, so it's hard to keep up with them. But the influenza, it's hard to keep up with, but we are able to actually make vaccines that stop most of the strains of the flu. But if people go to the doctor when they have a cold and ask for antibiotics, the doctor is supposed to say no, because number one, antibiotics will do nothing to cure a cold, and number two, if I give you antibiotics when you don't need them, that will speed up all of the bacteria in the world becoming resistant to those antibiotics. So, doctors are supposed to say no when you ask for antibiotics when you have a cold. Sometimes if you have a really bad cold or a really bad flu, then you end up getting a bacterial infection of your lungs, which is secondary to the flu. All right, so you end up having so much fluid in your lungs that bacteria will grow in your lungs. Okay, so antibiotics actually do nothing to kill a virus. Sometimes you get a bacterial infection which is secondary to the virus, in which case you would have to take bacteria. But technically, if you just have a cold or the flu, the doctor's supposed to say no when you ask for antibiotics. People sometimes try to take antibiotics, preemptively as a prophylactic to prevent a lung infection by bacteria, but you're not supposed to be doing that really. Now, the interesting thing about antibiotics is that many of them make you feel better because they're also a mild stimulant. But if you want a mild stimulant, then you can just take a strong cup of coffee when you have a cold. You don't need to take antibiotics. And the more we take, the more bacteria will become resistant to them. Now, finally, this is really scandalous. Many places in the world, farmers give antibiotics to their cattle in order to kill the bacteria, the symbiotic bacteria that are in the cow's digestive system, in the intestines, because the less of the cow, as food passes through the cow's digestive system, if bacteria are eating some of that food, the cows will not get fat as fast as they otherwise would. So there are some places where they actually give antibiotics to cows in the cattle food to fatten them up faster and save money. People are working it should be illegal, but for people who refuse to stop doing it, they are working for another way to make cattle fatten up faster, and that is to vaccinate the cattle against bacteria so that the cattle have fewer bacteria in their intestine and they fatten up faster, but it's not because you fed them antibiotics. All right, good example of antibiotic resistance and the rise of superbugs. The bacteria Staphylococcus aureus, which, as I told you before, is a pyogenic bacteria. It is generally, if you get a staph infection, you get antibiotics, and much of the strains of Staph aureus are resistant to antibiotics now. And usually, as a last resort, as a weapon of last resort to get rid of a staph infection, you use a very powerful antibiotic called methicillin. And if you have Staph aureus that's resistant to methicillin, then you have no way to stop it, right? So this graph shows you the rise over the years of methicillin-resistant Staphylococcus aureus, which is better known to us as MRSA. So whenever you hear somebody talking about MRSA on the news, this is what they're referring to. And so what's happened here is that if people went to the hospital with a staph infection, they would check to see if the staph infection was resistant to methicillin. And in 1990, you can see that probably about 2% of the staph infections that you had in the hospital were resistant to methicillin. And look at it now. This graph only goes to 2002, so it's 18 years old, but by 2002, 46% of the Staphylococcus aureus infections were resistant to methicillin. This graph is probably still rising. I haven't seen an update to it, but probably now we're up to some point where probably 70 or 80% of the Staphylococcus aureus is resistant to methicillin. So you have to use even more powerful drugs and the more powerful the antibiotic, the harder it is on the patient. So we're rapidly running out of antibiotics that we can use to kill bacteria. We have to start looking for other ways to kill bacteria. And there are other ways. There are other chemicals that people are doing research on, and you can vaccinate people against certain types of bacteria. But in the meantime, all of us regular people should not be asking for antibiotics in response to a cold, and our doctor is not supposed to be giving us antibiotics in response to a cold or a flu. So to summarize what we talked about the different types of cells that comprise the immune system. We talked about what they did. We talked about the different functions of them. We talked about the difference between the innate and the humoral immune response. We talked about the effects of chronic inflammation and fibrosis. We talked about how vaccines work. We talked about the network of vessels called the lymphatic vessels, and we talked about problems with the immune system. All right, our next lecture will be on the respiratory system.